In this video, we continue to discuss about the infectious agents in the topic microbiology. In the last video, I already discussed about the prion. Now, in this video, I will discuss about plasmids and bacteriophages. Now we come to plasmids first. First of all, we must understand what is plasmids. Let's suppose this is we have one bacteria. We have one bacteria. One bacteria. So normally this bacteria is it doesn't produce any disease. Okay, but what happened? For example, inside this bacteria, it has or they have what? Genetic material, and this is the gene or this is the chromosome of the bacteria. What we call chromosome or DNA. Yes, what we call DNA. Of course, within the chromosome we have DNA, and bacteria have DNA. Then this is what I'm talking about. This is a normal bacteria. It doesn't have capacity to produce the disease or infectious disease. But and what is plasmids? Inside this bacteria, we have one more like this. This is extra chromosome. This extra chromosome, what we call plasmid. We call plasmid. So plasmid is the extra chromosome of the bacteria. So when the bacteria have the plasmids are present in the bacteria, now this bacteria have a capacity to produce the infectious disease. This bacteria first, what is plasmid? Plasmid is a extra chromosome, but it has one capacity or it is capable to express itself. When it expresses itself, and then it means it can make copy by itself, and then it can make more extra chromosome. Yeah. When it make more, when extra chromosome, when it expresses itself, and then it become more, and then it become more dangerous. So the bacteria has the plasmid inside, and when it's multiply, and it's become very dangerous bacteria. Okay, but one more thing, and this thing is very dangerous. What is that? For example, I have another bacteria here. Second, and I have another bacteria here. This is also normal bacteria. Normal bacteria. This is the chromosome. This is the chromosome, genetic material. This bacteria, this plasmid, when it have a chance, it can, it can release their chromosome, their extra chromosome. It can release their chromosome. Let me make another color, then you can understand. So from here, from the chromosome here, and it's released to another bacteria. As I said, this bacteria is a normal bacteria. It doesn't have ability to produce the disease. This bacteria is also the same. This is 1, 2, and 3. Okay, so now in the bacteria number 2 and the bacteria number 3, when the plasmid from the bacteria number 1 is released to number 3, okay, you trust me that it has a capacity to release, and how it's released, then we will discuss in another video. But in this video, we just learn basic, or we need to understand the basic concept of the plasmid. Okay, now this bacteria also have plasmid, and this bacteria also have plasmid. Now you see, it's become more dangerous. The condition become more dangerous, and it's become the infectious disease. It is higher level than this. Okay. Now, if surrounding this bacteria with the plasmid inside, and then it can transfer to many other bacteria. Okay. So that is plasmid. One more thing, this is a very important thing. If you are a doctor, then you must know. For example, is this plasmid related to medicine? When you take the drug, when you are a doctor, then you give the patient the drug. What drug? Like When you treat bacteria, then you use the antibiotic. The drug is antibiotic. Okay, so like you give the patient or you give the bacteria penicillin G, for example, penicillin G. When you give penicillin G to destroy the cell wall of the bacteria, this bacteria, and then what happened? This plasmid, the bacteria has plasmids. It has a genetic material. It has a gene. So the gene can produce one enzyme. The enzyme can, the enzyme of the plasmids can destroy or can cut the molecule of the penicillin G. So finally, if you use penicillin G for the bacteria, and that bacteria has plasmid, and this penicillin become useless. It's become useless. Okay. Now, let's come to bacteriophages. I hope you understand Plasmid. Very simple like that. Plasmid very easy, but we have another lecture for the plasmid, how it's transferred. Their plasmid to other bacteria. Okay, now we come to bacteriophages. And what is bacteriophages? Bacteriophages, it is one type of viruses. It is one type of viruses. Virus have DNA. Now, virus has DNA. But remember, virus is not an organism. It is not organism. Why? Because whenever we talk about organism, so that organism must have cell. But virus doesn't have cell. Clear? Virus doesn't have cell. So that's why we are not consider virus as an organism. So bacteria is an organism because bacteria has cell. Now, what is bacteriophages? It is one type of virus. Now I have this bacteria. And inside this bacteria, we also have DNA of the bacteria. Is bacteria. Now, this is the normal bacteria. It doesn't have capacity to produce the infectious disease. But here, what it is? On this bacteria, they have one bacteriophages. So this is the bacteriophages. One type of virus. This virus will attack on this bacteria. Yeah, it's attached on this bacteria. It's come and sick on this bacteria. And then what happened? It has. Bacteria also have the DNA. DNA. So remember, bacteria. This bacteriophages we call one type of viruses, and then it has DNA. Remember, virus has DNA. All virus have RNA. Remember RNA. So virus doesn't have DNA and RNA together. No, it has only DNA or RNA. Now bacteriophages is one type of virus. It has DNA. And when it has a chance, it's attached on this bacteria. When it has a chance, what happens? If we make a hole, here, okay, and this hole, then they inject their DNA to this. This is the DNA of this virus. Okay, inject this DNA. So this DNA have gene. And this gene we call tox gene. What we call tox gene. So this tox gene has capacity or ability to express itself. Okay, when it expresses itself, then what happens? This bacteria has ability to secrete one type of protein. It's one type of protein. Okay, this protein. This is a special type of protein. We call that a special type of protein. So this protein, and this protein is a agent to produce the disease. And this protein what we call diphtheria. So diphtheria, what we call diphtheria exotoxin. This protein we call diphtheria exotoxin, and this toxin, clear, it can produce the disease. It means this bacteria can produce the disease. Okay, can create the disease, and this diphtheria exotoxin will produce one type of disease, and that we call diphtheria. What we call diphtheria. So this is also the one type of disease. It is produced or created by the bacteriophages. Now, I hope you understand what is plasmid and what is bacteriophages. Then, thank you so much.